Okay, maybe too easy. So I'm happy to see the perfect answers. So today's lecture is about the life is digital. So we are going to figure out how our genetic information is copied and also stored and transferred. So DNA synthesis direction is from 5' to 3'. Shao, are you ready for recording? Shao, are you okay? No answer. Anyway, move on. Okay, first question was too easy, so I was happy too early. So the synthesis of a DNA is utilizing template, and then the template is the opposite direction of the synthesis. So template is from 3' to 5', and using that information, we make a copy, but from the other direction, from 5' to 3'. So we will see the chemical structure later. Okay. Now RNA synthesis is from N-terminal to C-terminal. Who gave that answer? Let's check.
I did. I misclicked the button.
Misclick.
Yes,
Okay.
sorry.
Okay, the DNA synthesis, RNA synthesis, the synthesis direction is the same, from 5' to 3'. Okay, move on. Okay, who made mistouching again? Who is the number, the 5' to 3'?
I gave the answer.
Why do you do that? Any special reason?
I was just, my brain just stopped.
Okay, you need a coffee?
No, it's okay.
Okay, in this case, if I ask you, what is a template direction? So protein synthesis, the template is RNA. If I ask you the template direction, what would be your answer?
Could you please say it once again?
This question right now, this is a direction of a protein synthesis, right? Protein synthesis template is a mRNA, messenger RNA, correct?
Yes.
Then what is the template direction?
5' to 3'.
It is from 5' to 3'. That is important. That's from 5' to 3', not the other way. DNA synthesis case, the template direction was 3' to 5'. For protein synthesis, the template direction starting from 5' to 3' direction. Okay? Okay, perfect. At least you know what is the chemical structure of DNA. That's good. Now all of a sudden this became the difficult. Who gave the answer of sulfur?
Me.
Doyon, it was you.
Yes.
In protein, do you have any other example of a protein which has a phosphorus?
No.
To my knowledge, at least the 20 basic amino acids we don't have phosphorus in protein. Protein, we have a sulfur, nitrogen, but we don't have a phosphorus. Okay, move on. Okay, there is no fixed answer for this, but I guess you have heard about this comment from newspaper or some other part. So the question is, how many different kind of genes we have? Gene is the information to make the protein. Then this question can be like how many different protein we can make using our genetic information. There are many modifications can happen to the protein, so-called post-translational modification. If you count them all, maybe it's more than a million. But the basic sequence of the protein, or basic sequence of mRNA, or basic sequence of a gene, after we finished the genome sequence of the human, we found that's between 20,000 to 30,000. It's a surprisingly small number than we expected. Okay, so gene number, let's just say bacteria, simple bacteria is about 3,000, and yeast cells is about 6,000, and Drosophila, the fruit fly, is almost 20,000. So we believed our body is much more complicated, but surprisingly, total gene number is not so many. It's about 20 to 30,000, okay, at least by the DNA sequencing. Next problem, you need to remember this number and need a little calculation. Post-second students, you are good at calculation, right? Let's see. Okay, you are quite good at calculation, but there was a wrong number. So if you say the average gene size is about 1,000 base pair, so that will give you about 30 kilodalton of a protein. So that's kind of reasonable guessing for gene size. So if you have 30,000 gene, then our genome size should be about 30 million. That's the right calculation. But real length of our genome is 100 times bigger than that calculated number. Okay, so our genome is about 3 billion. 30억 base pair is the size of our genome. So that's why among those 3 billion, only 1% or maximum 2% of those sequences are allocated for genes. So that's why only 1–2% of the DNA is encoding protein structure. the other 98% to 99% of all other DNA sequences is we don't know exactly what is their functions, what are their functions. That's why for some time we call them as a junk DNA or some other names. Now we are learning more and more about those functions of other genes. Some weeks ago, we mentioned, we have some virus structure in our DNA, and that amount is almost 10% of total DNA. Okay, so protein encoding traditional or conventional gene is only 1% or 2%. Okay, it's a good time to think about that point. I think this is the last question. Oh, one more. Oh, one more. One more question. Okay, still more than half of you got the right answer. So this part we are going to discuss in today's lecture. And the today's lecture's title was Life is Digital. So we'll see how those digital structure is composed of. Okay, we move to today's lecture. Any questions so far? We have one. Okay. Any question? If not, we move to the lecture. Okay, today's the lecture, mainly I will teach you some of those basics of DNA and also translation, transcription, and RNAs. Okay. So we have many different kinds of life forms on our planet, but surprisingly, all of them, including the human and also very simple life form of bacteria or even virus. So all of them, we have a very common structures, so-called DNA. So far, we didn't find any exceptions. All the life forms that we found here, every life forms, they are utilizing DNA as a genetic information. Well, maybe a small exception as a RNA, but DNA, RNA is a cousins. They are very similar to each other. So utilizing this DNA, we make temporary information, so-called mRNA. So one mRNA is one information to make one protein. So this is a partial information of the DNA, and this process we call transcription. So if you call whole genes together, one gene is we call gene, all the gene together we call genome. So one mRNA, it can be transcript, we call. If we combine them all, we name it as a transcriptome. So anything we put "ome" at the end, it means whole collection of that the materials. So DNA make mRNA, and then eventually utilizing this mRNA sequence, and then using this machinery, this machine, you know the name, right? This machine is a ribosome, utilizing big subunit and small subunit, and utilizing another of the RNAs, so-called tRNAs. And this is rRNA, mRNA, tRNA. Three, the conventional other major RNAs are here. Utilizing these three RNAs, we make proteins. These proteins in the beginning is just a linear structure, linear amino acid coupled structure, and they need to be folded properly to have the proper three-dimensional structure and also functions. So last time we had some helper to make this kind of proper folding of this protein. What is the name of the helper? Anybody?
Chaperone.
It was a chaperone. Okay, chaperone is another kind of a protein, and that protein helps to fold this linear structure into this folded structure. Now, only information we transfer to our offsprings, for our babies, is only one set of information stored in DNA. That's all, basically. That is what you call genetic information. That genetic information store basically information to make these proteins. So that means we transfer only 20,000 or 30,000 blueprint of making the protein. That's all we just give to our sperm cell and egg cells. And by reading those informations, we build this machinery or building blocks of our body. And then those 20,000 of proteins should do everything in our body, starting from the one fertilized egg cells, growing up to the whole body, and then maintain all the functions in our body. And dating, mating, reproduction, everything should be controlled by these proteins. So if those proteins are doing their job perfectly, then we keep healthy and harmonize the status. If anything is wrong, maybe infection invaded by coronavirus or maybe other bacteria, that's one problem. Or sometimes our own proteins, if they have a slightly changed structure, so-called mutation happens. Or even though the mutation is not there, just a copy of this protein number is changed. Maybe more numbers of a copy, or maybe low number of a copy of a protein can break this balance. Then we have disease status like this. So this is basic the structure of DNA to the protein function. And one of those problem was, the question was, what is genetic material? Maybe we already know the main genetic material is the DNA. It's very obvious the answer for now, but just 1 century ago, even maybe half a century ago, it was not very clear. So then these two guys, the famous the Watson and Crick, these two guys suggested structure of DNA and suggested, they didn't say clearly whether DNA is the genetic information in the first paper, but in this paper, the first sentence was, DNA is a very interesting molecule. Well, DNA is quite boring molecule. It's just a sticky and only 4 different kinds of building blocks. And in comparison, protein is much more interesting molecules. They have 20 different building blocks, and they can have many different structures and many different functions. So naturally, century ago, about 100 years ago, scientists believed maybe rather than DNA, protein is much more interesting material, and it should have some important information to be delivered to the offsprings. That was the hypothesis. Now these two guys pick up that hypothesis and then suggested a totally new view, and this view was proved to be correct. And those DNA structure is here. Maybe you have seen this kind of structure before, but let's review again. So starting from this, the 1 unit of DNA is here. 1 sugar, so-called deoxyribose. 5 the 5 membered ring, and also 5 the carbon. 1, 2, 3, 4, 5 carbon sugar. Glucose is a 6 carbon sugar, right? So this is a 1 small, and then base. And then this 1 unit is connected by phosphate group. And in DNA structure, there is A, T, C, G, only 4 different kinds of building blocks. That's why even Watson and Crick at the time, they knew inside here, there is a fixed ratio of AT and GC, and there are 4 different kinds of building blocks. To be genetic information should have every character of our life. Then it should be very complicated, they believe. But these very boring molecules, very simple molecules, they should carry all those characteristics. It's really amazing, and also it is difficult to believe. Anyway, when they look at those structure, now this is a DNA structure. Now, if you look at closely this DNA structure, interestingly, there are only pairing A with T and G with C. There is a fixed ratio given that early times they recognized. And then this ratio is the fixed. And also it's found that from the X-ray structure of the DNA, they are facing to each other. They make a pairs like this. This is a very famous Watson-Crick's the DNA, the pairing structure. And then by looking at this structure, if you look at those the size, now these two bases are the parallel to each other. And then this length is about the 0.3 nanometer. So this is the 0.3. That means they make a double helix, you ever heard. And then 10 ton will be about 3 nanometer size. Okay? 3 nanometer, 4 nanometer is about the size of membrane thickness, you remember. Now this diameter is about 2 to 3 nanometer size. It's relatively thin. And there is two growths. One is minor growth, and there is a major growth. Major growth is this open site, and minor growth is near to this carbohydrate structure. Ribose is a little more bulky, so this is a little shallow, and then this is a wide open growth there. And if you look at those, so before we move on, based on these informations, let me give you one quiz. Now in the quiz that we said, our genome has a 3 billion base pair. Okay. Now another hint is, let's say carbon-to-carbon bond is approximately the 0.15 nanometer, 1.5 angstrom. And if you put our DNA in one line long, okay, then what can be the physical length of that of our total DNA? Can you calculate quickly? Let me give you 1 minute. Anybody got the answer? If you get this kind of questions, you can look at this structure and then maybe count those bond number between one unit to the other unit. How many bond here?
4 to 5 bonds.
Is that 4 to 5? It's not difficult. You can count the exact number. From one unit of DNA to next unit, the bond length, the bond number, how many bonds?
5 bonds.
Does it look like a 5? Everybody has the same number? Let's count together. Who said 6?
I said 6.
Sorry, I don't see your face. Was it Juan?
Yes, I did.
Okay. So let's count together. So starting from this P, 1, 2, 3, 4, 5, 6. From one P to the other P, it's 6 bond. That may be shortest path, isn't it? If you go here this way, that's longer. So this should be shortest path. If these bonds are 6 bonds are zigzag structure, then approximately we can assume this bond length is approximately 0.1 nanometer. If you assume so, then what can be the total length of our DNA? Come on, it should not be very difficult.
3
Billion
meters.
is sorry. Don't say again.
1.8 meter.
1.8 meter. So billion is a 10 to the 9th. nanometer is a 10 to the minus 9, then they can be canceled out. Then one is the 1.8 or 1.6 nanometer, and the other is the 3. Then if you multiply them together, and that's 1.8 meter. That's approximate the length we can assume. So our DNA, each cell has one DNA. So if you take out the whole DNA and make one line, that length is approximately 1.8 meter by using this kind of assumption. But if you use a different information, let's say, in the previous page, we said interbase distance is approximately 0.34 nanometer. That's experimentally measured. If you use this information, then what is the length? Hey, come on, it's not a difficult question. This information is given by that X-ray structure. So this is the experimentally confirmed the length. And if you use this information, previously when we counted this, we used the 0.6. Now we use this one, then our DNA length is
1 meter.
1
it's
meter.
about 1 meter, right? So experimentally, that's about 1 meter. And by utilizing this kind of simple calculations, simple the assumption, and still that's kind of just a twofold the difference. It's close enough. One is 1 meter experimentally. By assumption, that's about 1.8 meter. So our DNA size is that kind of length. So you don't want to say that's distance between from Earth to Moon kind of things. Okay. If you combine all cells DNA together, maybe that's much bigger. But one cell of DNA of a human is about 1 meter or 2 meter. So it's about height of the physical height, right? So that's approximate number. Now, this 3 billion, when we say 3 billion base pair, this is counting 1N or 2N. What do you think? When we say our genome, human genome is 3 billion, then do you think it means that's 1N count or 2N count?
Oh.
I didn't hear you. Say again.
When
Youngjun,
2N.
it's a 2N you think? Now, 1N versus a 2N. Everybody, we have the 2N structure. Majority here is we have only exception. Only one over here, the 2X, and all others we have X and Y. Now, if we count other than those X and Y chromosome, all other 22 pairs of genes are exactly the same, right? Then when we say human genome is a 3 billion base pair, do you think that's really double count of those two?
Oh, there's one.
No, it's not. So actually this 3 billion is 22 pairs, just one each, 22, plus X plus Y. That's about the 3 billion. Okay. So when we did the genome sequence, whose genome should be sequenced? So that's also the big question at the moment. So the first genome sequencing the work, Human Genome Project, about 20 years ago, they mixed 10 people's the DNA together, and there should be some, half of them is the male, half of them is a female, and there's a mix of X and Y, and those mixed sequences and average sequence, those sequences are about 3 billion. That makes sense, right? So that's why if you take out the DNA from one cell, it contains the 2N. That's why if you calculate it here, that's about 1 meter size. Then the 2N should give you 2 meter altogether. Okay. Now this is those total lengths of our the genome. Now this is the building blocks of DNA. So now here we have the sugar, and 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So 5 membered sugar and 5 carbon sugar, and there is no hydroxy here. Ribose has a hydroxy here. That's why this sugar is called as deoxyribose. And these building blocks, as you can see, all of them has a 3 phosphate. 1 phosphate here, AMP, is not energetically high enough. That's why this is kind of a stable one. When they have a 3 phosphate, this ATP is kind of activated the building block. So utilizing these activated building blocks, now the DNA synthesis happens from this side to the other side. So this is the building block. Chemistry happens by this 3' hydroxyl attack here, and then activated triphosphate is decomposed by removing this pyrophosphate or diphosphate, and then this phosphate bond can be formed. This is the direction of a synthesis. So even though you forgot about the direction of a synthesis, now you can see always the growth of this DNA, the strand, is starting from 5', always 3' attack here, and then connect to this direction. Because of this reason, so nature could have another option. Maybe this triphosphate could be added here, 3 positions. Then 5' hydroxyl can attack, and then it can go this way. But seems like nature decided this way to synthesize. That's why DNA synthesis always happen from 5' to 3' directions. And this direction, interestingly, this template direction is the opposite to this. Now, if it goes exactly the same way, we may call this is a parallel synthesis, but this is the opposite. Here, synthesis direction is from 5' to 3'. Template is 3' to 5' direction. It's anti-parallel structure. That's why the direction of this the synthesis is opposite directions. Now, if you look at those DNA replications, now first step is DNA is always carrying double strand, and then to synthesize a new strand, we need to open this DNA from double strand into the 2 single strand. You remember the enzyme name which opens this DNA strand from double strand into the single strand?
Helicase.
It's a helicase or gyrase kind of enzymes are involved here. Gyrase is making topoisomerase of the function. Helicase open up this part. And then one direction, let's just say this one is, this end is a 3'. Okay. Now synthesis can happen here. It can move smoothly, go this way, and then it can keep going over synthesis. So synthesis of DNA, originally it was a double strand, open up from one side. And then one strand can be synthesized smoothly from one end to the finishing to the other end, keep going. This strand does not have any problem, but this strand has a significant problem because original strand was this is 5', moving to the 3' this direction. That's why synthesis cannot happen from here to here. Synthesis should be done from here to these directions. Then, problem is this DNA is not opened whole structure, only one side is open. That's why synthesis cannot be finished by smooth the one strand. So one scientist, Japanese scientist, predicted maybe some fragment was synthesized. So this much, and then later it can be connected together to make the full strand. So following that scientist's name, this fragment is called as Okazaki fragment. And this was proved correct later, and he got the Nobel Prize by just doing this the prediction. But it's a very important prediction, and the experiment was done by somebody else. But using this information, he got the Nobel Prize. So that's why DNA are the replications. One side is one smooth synthesis. The other side is a fragment by fragment synthesis. Okay, so this is the DNA synthesis, but both of the strands, as you can see, the synthesis happens from always 5' to 3' directions. But template is opposite. Template here, this template is starting from 3' to the other side is 5' directions. By reading this sequence and then synthesize it happens. Okay, next is the DNA from DNA to the protein. In central dogma of the modern molecular biology, the first thing we found was starting from DNA. Always this DNA information is transcribed to RNA, and then utilizing this RNA information, we make a protein. So this was kind of a simplified, very hard the dogma. But later we found many more complicated situations, especially with those RNA-based viruses. And sometimes there is some RNA can be used as a gene, and sometimes it's a positive strand or negative strand. And these RNAs need to be incorporated into the host genome. That's why sometimes they need to make not only from the DNA to RNA, sometimes RNA to DNA. In this case, we need the different enzyme, so-called reverse transcriptase. And also sometimes the RNA polymerases based on RNA template into another, the RNA synthesis. Always this can also found. So the central dogma becomes much more complicated. For all of those cases, whatever the template is the DNA or RNA, always the synthesis happens from 5' to 3' direction. Template direction is 3' to 5' directions. Now, once we have this mRNA, now we make the protein. Protein building blocks are the amino acids. And here one side is amino, the other side is acid. That's why we call this building block as amino acid. And amino acid coupling, by utilizing this acid part and amino part, one molecule of water is removed. And then so-called peptide bond is formed. Amide bond or peptide bond is formed. This kind of water removed the coupling, we call condensation. And opposite reaction from here to this, what do you call? By adding water and you break up. This kind of reactions, how do you call?
Hydrolysis.
This is a hydrolysis, okay? By adding water and you break up, this is hydrolysis. These two reactions can be reversible reaction, but when we make this peptide, amino acid is a building block, and then they are coupled together and they make linear polymer of the structure. This is the first product of our gene. Now, by looking at these structures, now we can see this is the peptide bond in the middle. And synthesis direction is, again, we can think. Now, if you look at here, now this is nucleophile. This is attack these positions. So that's why the reaction happens from this side, always starting from the N terminal, and then growing into the C terminal directions, and add up one by one of these amino acids added. So synthesis of a protein is always happening from this side to the other side. And in this case, the template is mRNA. mRNA's template direction is 5' here, and then 3' here. Okay. So template direction is different from DNA or RNA synthesis. Now, based on this, we have 20 different kinds of amino acids we build in. Now, if you think about starting from the DNA informations, that information is transferred to protein synthesis. DNA is composed of only 4 building blocks, but amino acid has 20 building blocks. So there is a significant mismatching of those information. So if you think about computer systems, let's say our computer system is right now binary systems. It is composed of either 1 or 0. So if you think about those amount of information we can keep, maybe these binary systems we can divide 2 by 2 by 2 like this. Then 1 unit like this. this is 1 unit of the information. Then this is 1 byte. 1 byte is composed of a 2-bit. And this computer, we can call this computer is 2-bit computers. In this case, if it is a 2-bit computer, then 1 byte can carry the information of a 4. It can be 00, 10, 01, 11, right? So that's why 2-bit computer can carry only 4 information. If it's a 4-bit computer, now utilizing this 4 units, then how many information can you carry with a 1 byte?
16.
16.
It should be 16, right? So 2 by 2 by 2 by 2. So it should be 16 different kind of information we can get. So let's just say if our body is using this kind of binary systems, our genome can be scored in this kind of a binary system. Then to encode 20 different amino acids, 4-bit is not high enough. With a 4-bit system, we can designate only up to 16. So we need bigger system. How about the 8-bit system? If it's an 8-bit, then 1 byte, how many information it can carry?
256.
It's a 256. If you go further, if it's a 16-bit, now it should have a lot more informations. Then we can get more than 65,000 different kinds of informations. Maybe we may not need this high. Then if our information system, if our genetic information system is a binary system, what is the minimum size of bit we need to designate the 20 different amino acids?
5-bit.
It should be 5 of the bit. With the 5 bit, we can carry 32 different informations, right? So if our genetic system is a digital, and if you use this kind of binary systems, let's say if you use computer system to designate each amino acid we need, then we just carry this kind of informations, 5 of them, then we can assign all different amino acids. That's something we can imagine. But if it's a binary system, let's just say, this looks like the total whole mass like this, then how do you know which is starting point and which is the ending point? It's difficult, right? If you just cut magnetic tape in the middle of randomly, then it's difficult to see which is the starting point and ending point. So that's why usually in our systems, usually we use, let's say, if it's a total 0, then maybe this is a starting signal, maybe total 1, and this may be the ending signal, kind of thing we can assign. Then maybe similar things in binary system, if we use a 5-bit system, 32 information, but at least the one starting point and ending point we need to save. Then amino acid number wise, we can assign maybe 30 maximum amino acid we can assign, right? So similar things that we can apply to the biological system. Now we found that our biological system is not binary systems. We have 4 different kind of the building blocks. ATCG is similar to 0, 1, 2, 3. And utilizing this the quaternary system, and then it has also direction. It's not randomly from the either direction. It has direction because it's chemical structure and it is a defined one end to the other end. Maybe it could randomly we can assign, but anyway, later we found that the information flow is from 5' to 3' direction. Then to encode 20 different amino acids, then you may think, how many different how many are the bit we need to use from DNA or RNA systems? If it's just 1 bit, if it's just 1 bit, we have only 4 information we can carry. So definitely this is not big enough. Then maybe 2 bit is 16 information you can carry. Still, that's not high enough. If it's a 3 bit, maybe 64. If it's a 4 bit, how many is it?
56.
56. So the 64 is already bigger, the higher number than 20. But you know, who knows? Maybe sometimes we don't need the 32-bit, all of them. But sometimes we use a 64-bit computer just to carry the bigger information and also for the speed issues. Maybe, reasonably speaking, maybe they could predict that our system may carry 3-bit systems, but it is possible that nature have chosen maybe 4-bit other system too. So the scientists started to think how we can assign each of these sequences matching to different other proteins. Later, they found that yes, it was a 3-bit. Still, this is much bigger. Maybe 1 starting sequence, 3 ending sequence, removing the except for those 4 different sequences. Still, we have 60 different kind of amino acids we could assign. So that means maybe some amino acid has more than 1 coding. And is covering those 20, and maybe those the relative ratio of those amino acids may be different depending on those amino acid sequences. So later they found that this sequence is the following: 5' to 3' direction is the matching to N-terminal to C-terminal directions. So both of those are the 2 polymers, DNA, RNA, and also peptide. They have a direction, and this direction should be matched from here to here, like this. Okay. Now, how do you think, the first, the scientist who discovered this sequencing, whether this AUG sequence is matching to which amino acids? What kind of experiment do you think they have performed? If it is you know that there's maybe 64 different sequences, different combinations, AAA, AAG, AAC, 64 different kind of informations. And what kind of experiment would you like to do to find out which 3 code is matching to which amino acid? How will you design your experiment? To make it very simple, then you need to assign 20 different amino acids to avoid that complicated combinations. What choice would you like to take? Maybe simple idea, maybe you just use exactly same ones. Maybe you can use AAAAA. So U is equivalent, the RNA equivalent, the T. DNA carries the T, ATCG. RNA has a U. It's almost the same, except one methyl group. So these 2 scientists, so one was the professor and one was his student, and they made just a simple same sequence of RNA. Then where they start, it doesn't matter, right? Anyway, those 3 sequences should be UUU. And then they tried to synthesize the peptide utilizing this sequence, and they found the peptide was made as phenylalanine. So it's all fa was synthesized. Then the first coding was made. UUU is a fat, right? So this is historic, the first experiment, and then their notebook to make this recording of each sequence. So starting from this, maybe you can try AAA and GGG, CCC, and then mark those sequences, and then change one by one, and then try to make those kind of the matching. So this is historic the experiment, and this original lab notebook is displayed at the gate of NIH in US, National Institute of Health. If you enter those main building, so they displayed this historic notebook there. This is kind of following Watson and Crick's important discovery of DNA is a genetic information, and of course they got Nobel Prize. And Watson is quite interesting character. He made a really great discovery but he has a lot of social issues later, making some funny comment about the racial discrimination and also a little arrogant. And people didn't like him, some people. And then at one symposium, somebody asked him, hey, even though you got a Nobel Prize, you cannot say this kind of too big thing. And he mentioned, no. It is not a Nobel Prize. It is the Nobel Prize. So he was a so proud person. Of course, his work was so important. Now, utilizing that information and then making out this encoding table, translating of this mRNA sequence into amino acids. It's a very, very important information. Of course, these two people got Nobel Prize for this work. And then this is the coding, now we know. Starting from here, C-A-G, this make E, and U-C, whatever, the last three is not important. First the two decide whether this is a serine, kind of the table that could be built. And utilizing this information now, if we have mRNA sequence or DNA sequence, now we can have the sequence of, something happened. Okay, do you still see the 20 amino acids screen?
Yes,
Yeah.
can.
Okay, good. So now if this 20 amino acids is randomly distributed in our protein, then relative ratio of this amino acid should be 5% each, right? Then it should make 20 by 5, it should make 100%. But already we have seen in genetic code, it is not exactly same the ratio. Some of those genetic code assigns more amino acids than the other. Then also some of those amino acids is very simple, like alanine or glycine, but some of them looks like quite complicated. Tryptophan and some structure is quite bigger. So that means this kind of amino acids is maybe more expensive to make and maybe more precious. And you may have heard that some of those the essential amino acid, something we cannot synthesize in our body, then we need to take those kind of essential amino acid from outside. So that's why we can expect maybe those relative existence the ratio inside the protein may not be exactly the same 5%. Now, as we can expect, alanine is much higher contents. 5% is the average number. Alanine is much higher, and cysteine and tryptophan, where is it? Tryptophan, that's kind of much lower. So this kind of amino acid is more the precious and also more expensive than others. That's why relative ratio of amino acid distribution is not same. So animal and plant, both of us has protein, but those ratio of this amino acids composition is different. That's why some of those plant, the protein is not good enough to support the animal body. That's why sometimes it's necessary to have some protein from animal sources. Okay, let's think about the RNA. So DNA structure here, and this is the RNA structures. Almost the same, right? Now, DNA has ATCG, RNA has AUCG, and Watson and Crick base pairing happens only to this side. And difference between U and T has a methyl group here. U doesn't have a methyl group here. And Watson and Crick base pairing happens only this side. That's why this methyl group does not interfere of that Watson and Crick the base pairing. So that's why if DNA can make double strand, same sequence of RNA Do you think it can make double strand with the DNA? The Watson and Crick base pairing happens to this side. Phosphate is here. If you add the same sequence of DNA and then opposite sequence of RNA, do you think they can make a double strand? Never heard? They do. So if their DNA sequence and RNA sequence are matching complementary to each other, then they can make double strand. And funny thing is, even that DNA RNA double strand is a stronger binding than DNA. How about RNA to RNA double strand? Do you think it's possible? That is also possible. And the strength of a binding of RNA strand to the other RNA is even stronger than DNA RNA. So those kind of sequences is not common in our body, but there are some virus, they carry that kind of sequences. That's why if we encounter any kind of a double-strand RNA in our body, then our body is resistant to that and try to destroy it. That is kind of evidence that there is something funny material, potentially virus, infected our body. Okay, so double-strand the RNA is not common in plant or the animal body. Okay? One more thing. Here in this the problem, why DNA versus RNA, which is more stable?
DNA,
Why?
because of that hydroxyl group in RNA.
Can you explain a little further?
I don't know exactly, but that hydroxyl group was
Usually that answer is a good answer, but can you explain a little further? Why is it? Anybody? Captain, you try first.
phosphodiester bond is break by that hydroxyl group.
That's a very good point. So in this picture, let's just say here, hydroxyl here, this hydroxyl can attack this phosphate. And this phosphate is usually, this is a quite stable bond. Amazingly, this phosphate bond is maybe 100 times to 1,000 times more stable than amide bond or peptide bond. Usually in chemistry, the peptide bond is considered to be very stable, but this phosphate bond is even more stable than peptide bond. That's why in case the peptide cannot survive, still DNA, they can survive. That's why we can take out those genetic information from very old the samples. Okay. Now, if we have one hydroxyl group here, this hydroxyl group can attack this phosphate, and then it can induce cleavage of this phosphate the group here. So it's much chemically, maybe 1,000 times or 10,000 times more susceptible than DNA. And also, there are enzymes which can break this the bond. Chemically, biologically, RNA is very, very unstable compared to DNA. That's why DNA is utilized as our genomic information to keep the information very long. And RNA has been used to make temporary copy of this information. And by utilizing that temporary information, we make a protein. And the lifetime of this RNA is not so long. So after some time, maybe 20 minutes or 1 hour, this DNA is slowly degraded. That difference is huge. That's why most of our life forms, we use the DNA as our genetic information. But recently now we see some virus, like a coronavirus, they use RNA as a genetic information. Because of this instability, they have a very good chance to have some changes of those sequences and structure. That's why their mutant rate and the evolving rate is much faster than DNA-based virus or other life forms. So they chose different strategies. They selected an unstable identity. That instability gave them higher chance of changing their structures. That's kind of a paradox. And also, they need to get a good balance. If not, their genetic information is too unstable, then they cannot survive. And they needed to have some protecting groups like protein code. See? There is one more problem. We just covered about the DNA and RNA. Now, we have always the one pair of parents, father and mother. If you want to follow only father side, which structure or which material should we look at?
Y chromosome.
That's a very good point. Y chromosome. Because mother does not carry Y chromosome. Mother carries the two X chromosome. So those egg cells carries only those X chromosome, one of those X chromosomes. So it doesn't have any Y chromosome. Y chromosome is only through father to son. Only male side, it goes down. That's why if you track those genes in Y chromosome, then you can separate those information, whether this is coming from mother or coming from father. Okay, Y chromosome gives you the idea of whether this is from father side genetic information transfer. Then how about your mother side? Then only you can do the father side. Million years ago, let's just say there is some Lucy in Africa, Australopithecus. So we call her as kind of a common mother, not common father. Then how do you know whether that information is connected through father side or mother side? If you want to know those mother track, do we have any option? X chromosome is not right answer because X chromosome can be transferred both to mother to son, father to son too, father to daughter too. So X chromosome is not right, not a choice. Is there any other option? Sorry?
Mitochondrial DNA.
Yep, that is a good answer. Mitochondria. So when sperm cell and egg cell meet together, we only count those two nucleus meet together and make 1N to 2N. But when they meet, sperm cell does not carry mitochondria. Only egg cell carries mitochondria, and there are a lot of different proteins there too. So for one fertilized egg cells, if they want to survive and grow, they cannot just start from two genetic informations. They need some helpers which can translate, transcribe those genetic information, and also some helper over energy generation by mitochondria. So that's why the mitochondria is provided only by eggs. So mitochondrial information, if you follow, then you can track those mother side genetic informations. Okay. Now, let's think about the transcription. Transcription is making mRNA from DNA. Now, here is one strand, 5' to 3' strand. The other strand is a 3' to 5' strand. Okay, they have a matching opposite complementary sequence to each other. And when this mRNA is synthesized, now here upper strand and lower strand, two strands, right? Upper strand versus lower strand. Which strand represent genetic information?
Lower than.
Do you think this is a lower sequence is the genetic information?
Upper sequence.
Upper.
It's so funny, right? The sequence of genetic information, we just look at those sequence of 3-bit information to make protein. That information sequence is this mRNA sequence. Okay, that mRNA sequence you should start to read from 5'. When they make this mRNA, they do not use this genetic strand. They use opposite strand as a template and make this mRNA, right? When we read DNA sequence and when we count, okay, this is one gene, okay? We found those starting point and the end point. So this is one gene. Then we read this sequence as a one gene, okay? Upper strand is a genetic information. But the synthesis of mRNA, they do not use this sequence. They use this sequence as template and then make mRNA. Do you follow me? Now here, those questions said, and if you call this sequence as an mRNA sequence, this one is kind of different sequence, right? Then if this makes sense, this is a sense strand. mRNA strand is matching to protein sequence. That's why this is a sense strand. Then the other strand here, this should be called as an anti-sense strand, right? And further, this one may be anti-sense, or this is a real sequence, and then this is a complementary matching sequence. And this one can be called as a complementary sequence too. And in RNA case, we had a positive strand and negative strand. Positive strand means that's a RNA virus. That positive strand is matching to this mRNA. That's a positive strand. That positive sequence is matching to protein sequence. Negative sequence is a complementary sequence to that. Okay. So one of the quiz I gave you is one side, sense strand, and positive strand is this side. And the other side, anti-sense strand, complementary strand, or negative strand should be this side. mRNA should go to where? Left or right? mRNA should go to left or right? Positive strand, sense strand, anti-sense strand, complementary strand, negative strand. mRNA should go to left or right?
Left.
It should be left. This is mRNA, right? mRNA sequence is a sense strand. Okay. Now that's why this DNA, this side. So let's say this is some genetic information. So we have two ways to make a genetic information maybe exactly the same copy. Then positive to positive. Or we make this the template, utilizing this as a template and make a copy. Then if this is information of the mRNA, we call this the sense strand. And this side is maybe complementary or anti-sense the sequence, right? Now in this case, if this is mRNA and template of this DNA, we call complementary DNA or cDNA. Okay, so we'll hear that term sometime soon. cDNA is template of mRNA. So if you have cDNA, if you transcribe mRNA, then what do you get? Do you get positive strand or negative strand?
Negative.
Think again. This is complementary cDNA. This is the cDNA. Okay, cDNA is a complementary antisense. If you make an mRNA from that, that is a positive strand. That's a little confusing. That's why I point out this part. If you wanted to make an mRNA, the sense strand, we needed to have a complementary DNA, cDNA. That cDNA is not genetic code. It's a template of genetic code. Clear? You should think this repeatedly. This is very important. Then later we can utilize this for the applications. Okay. One last picture here. Here, this is a DNA strand. This is a DNA. Now each line here, these guys are RNAs. And small dots here, those are ribosomes. So that means a long line of this DNA, RNA synthesis can happen many copies at the same time and moving from one end to the other. Even before they ended those mRNA synthesis, protein synthesis can happen together at the same time. Now, if I ask you a question, whether this end to this end, which is the starting end? Which is the starting of transcription? Left side or right side?
Right,
Because
because the left side ribosome is have many, and that RNA strand is longer.
yeah, RNA strand is longer. That's a good answer. So start here and then slowly it is moving and then RNA is growing further and further, right? That's why this is beginning site and this is the ending site. That means DNA here, this DNA is a 5' or 3'?
3 prime.
This should be 3' because the synthesis should happen from 3' to 5' directions, right? By the way, this transcription should happen inside the cell, inside the nucleus. Protein synthesis should be done in cytosol, outside of the nucleus. How come they can happen together?
In case of prokaryote.
This is a bacteria. There is no nucleus. That's why you can see this kind of things happening together, or maybe in vitro. Okay, today's mainly I tried to give you the lecture because there's a little too much information here. So I tried to deliver some the basic informations. I saved the materials for the DNA sequencing, including PCR and DNA sequencing kind of things. So that part we are going to cover on Thursday. And the deadline of the assignment, there will be the assignment 6, right? Tomorrow. And same way, hopefully you can send me the on time by tomorrow morning. Then we decide several important the talks for Thursday. Any question for today's lecture?
Professor.
Yep.
When I check the debating form that suggests select the subject of the presentation.
Yeah, I found you selected the same one with Thomas, maybe.
Yes, I saw it too. So I wanted that it is okay to do the same subject as him.
So who said the transcriptomics? One said the transcriptomics, one said the binary 교수.
I did the transformic, and the other did professor.
Okay, so basically those are the same the same topic I found. One way is, Dongwoo, did you finish your assignment?
No, I'm still working on it.
Not yet. Juan, have you started?
I almost finished it.
Which part did you focus on? Nanopore technique or the transcriptome itself?
Actually, I'm focused on the computer calculation in her the professor's essay.
Okay, if Juhan already has moved that way, then Juhan, why don't you focus on the computational part and the meaning of the binary 교수's the paper? Why it is important kind of things.
So
Okay.
so I need to present it on the the
Depending on your material, I will decide. And
Yes.
Dongho, why don't you focus on nanopore technique itself?
Oh yes, I didn't know that my content was the same
not-
with other person.
It's not your fault because you picked up first, so that's not your fault. So I didn't intervene.
Well, actually, I didn't do it earlier than Juhan, so
Is it
I
Juan did
guess.
the first?
yes, I did first.
Okay, sorry. I thought I was confused. Dongho, then why don't you focus on the nanopore technique itself?
Okay,
Nobody
okay.
chose
I'll
that
change
part.
to that.
Okay,
Okay, yeah.
it's a part of that. It's a part of that part. 김빈내리 교수님 paper utilized that nanopore technique. I put up some material in the video clip, but I think it's an important technique, so it's worth the trying. So compare those nanopore technique with the other NGS techniques. Okay, that's a Dongho's.
Okay, I'll work on that.
Okay, good. Any other questions? Okay, if not, okay, let's finish today's lecture here. Okay, thank you guys.
Professor.
Thank you, Professor.
Professor.
Thank you, Professor.
Thank you, Professor.